This lecture covers material found in Chapter 6 on communication integration. It also has a small little bit of material that's found in Chapter 1 about homeostasis. The body communicates from one cell to another cell in a couple of different ways. We have what's known as local communication and long-distance communication. Local communication is, as the name implies, going to occur in cells that are found in a centralized nearby area. We're not talking about a systemic communication, something that's immediate or local, right? And there's three ways that this can happen. We can have local communication that occurs via gap junctions, in which you have two cells that are connected to each other, so their cell membranes share gap junctions in which we can pass chemicals directly from one cytoplasm to the other cytoplasm, so we can communicate via proteins, hormones, those types. We can also have what is known as contact-dependent cells. In this case, again, you have adjacent cells. However, there isn't a porous membrane. The membranes are separated, but they do have protein, integral protein membranes and receptor proteins that are found on the cells that can then bind to each other and elicit different reactions to occur. And then the last way we can have local communication by neighboring or joining cells is by the release of proteins, what we call autocrine. So these are autocrine signals. We create a protein molecule, it gets secreted, and then it can act either on itself. So if this protein binds to a receptor on the same cell, then it's called an autocrine. If it, however, binds to a receptor on a neighboring cell, then we would call it a paracrine. An example of a paracrine would be like histamine, right? You injure a cell. So let's say you get a cut, that cell starts to express the gene for histamine and produces histamine, which gets released, and histamine can then bind to histamine receptors on nearby cells that tell them, hey, look, there is actually injury in this area and we should start the inflammatory response. An example of an autocrine cell may be like interleukin-2, which is a gene or is a protein that gets produced by T cell lymphocytes. And when they start releasing that, it tells that T cell it's time to mature and to develop because there's an immune response that's needed. So these are the three ways that we can have local communication: gap junctions, contact-dependent signals, and then having autocrine and paracrine signaling. Now, if we're talking about cells that are much further away, here we're talking about things that resuscitate long-distance communication. And there's two systems that deal with long-distance communication. The first is going to be the endocrine system. And the endocrine system functions such where we have endocrine cells that produce hormones that get then secreted into the blood system, will be circulated, and then bind to cells that have receptors. So if a cell doesn't have a receptor, we get no response. If there is a receptor, we would then elicit a response. But this is a systemic system, right? This cell could be very far away from that cell, thus long-distance communication. The other mode by which long-distance communication can be accomplished is the nervous system. So in the nervous system, we now have a cell signal that is sent by a neuron, and the neuron releases neurotransmitters that then bind to receptors on target cells, and we have a response. And in some cases, we have neurotransmitters that can get released into the bloodstream, which we call those as neurohormones, and they function similar to like an endocrine system where they would be then circulated by the blood itself. Most nervous system responses are done through neurotransmitters, and you can see it's very specific. That neuron goes to this target cell. Whereas the endocrine and the neurohormone system is relying on systemic circulation and only the appropriate cells with receptors having that response. So the type of long-distance signals that we can have In the endocrine system, those would be hormones, and in the nervous system, it's either going to be accomplished via electrical signals itself, so changes in membrane potential, or what we call neurocrines. And there's 3 types of neurocrines that we can have. We can have what are called neurotransmitters that get released from the neuron itself. We can have neuromodulators, which are compounds similar to a neurotransmitter, but rather than eliciting a response, they can alter responses that occurs, or neurohormones that get released from a neuron but then circulated in the blood. So transmitters and modulators don't get circulated in the blood, only the neurohormones do. Now, this begs the question, why do some cells respond to a chemical signal and others don't? Why don't we get these kind of, why don't all cells respond to these signals? And I've kind of touched on this a little bit already, in the sense that there are specific receptor proteins. Those receptor proteins have a specificity that will only bind to the appropriate chemical signal. And so by this method, we can then have specificity in our reactions and our signal pathways. Now, a signal pathway all pathways have these basic steps, right? There's going to be a signal molecule. It's going to bind to a membrane protein, and that's then going to activate an intracellular signal response within that cell. That response then is going to alter the proteins that get produced by that cell, and that will result in a very specific response. We call these signal molecules ligands. Okay, a ligand is a signal molecule that binds to a receptor protein. Okay, sometimes we'll also refer to that as the first messenger. Once we activate, sometimes we have intracellular signal molecules. This would be a secondary messenger. Right, so it's something, a secondary signal that gets released inside the cell. Much like how we send signals when like radio waves, or when we're sending communication signals, there's some processes by which we have to activate and alter the initial message. So like when we send a radio wave, right, that radio wave gets sent out, it gets received by the antenna on your radio. All right, and then that radio then converts those radio waves into sound waves. And inside the radio, then we also have an amplifier that would then increase the strength of that signal to where we would be able to perceptively hear that. So there's 2 things that occur. We have the transducer and the amplifier that make that signal have the response that we ultimately want to have. So in a physiological pathway, transduction also occurs, right? We have this extracellular signal. It may be in the form of, say, a neurotransmitter, right, that binds to a receptor. That neurotransmitter doesn't necessarily mean anything. That the binding of that neurotransmitter at the receptor then has to be transduced into some type of message that then the cell can respond to. And that usually happens through secondary messenger pathways, in which we have this cascading of additional proteins that get activated or inactivated, that then change usually transcription and translation of new proteins within that cell. And the amplification process that occurs when, let's say, we have a single ligand bind to a receptor. That single ligand binding can actually sometimes turn into multiple secondary messengers being produced inside. So small response, but then it's amplified inside the cell, usually by amplifier enzymes. So this binding at the receptor turns on an enzyme that then activates lots of other proteins. All receptors exhibit those same properties that we talked about last time of saturation, specificity, and competition. So, as an example, here we see norepinephrine. Okay, norepinephrine is a neurotransmitter, and there is a molecule called epinephrine, very similar, right, in which we've just changed the stereochemistry slightly. Instead of this being instead of us having a single NH2 here, we have a nitrogen, and then we have an additional carbon that's bound onto it, right? So the addition of a single carbon makes the difference between norepinephrine and epinephrine. Epinephrine is a neurohormone. It gets produced in the adrenal gland, in the adrenal cortex, and it's circulated in the blood. Okay, both of these can bind to what we call adrenergic receptors. Okay, these are receptors that have a specificity that allow both of these compounds to bind to it. That being said, certain adrenergic receptors may be more, their specificity may be more inclined to allow norepinephrine to bind than epinephrine, or vice versa. We see that in that we have two different isoforms of these receptors. We have what's called the alpha and the beta. And the alpha is going to favor norepinephrine, the beta receptor is going to favor epinephrine. Okay, and this would reflect how we have actual competition, right? We can have both of these compounds being present, but because of the specificity of that receptor, competition can favor one versus the other. Additionally, we can have other molecules, other compounds that can bind to those same receptors. So we call anything that activates a receptor, any compound that can bind, any ligand that can bind and activate a receptor, we call that an agonist. Okay, if you have a compound that can bind and then block that receptor to where we get no response, we call that an antagonist. So there's competition between agonists, and we can also have competition between agonists and antagonists. In addition, some of these responses can lead to what we call up and down regulation of receptors. Up regulation is where we'd increase the number of receptors on a membrane. Down regulation would be where we decrease the number of receptors on a membrane. And that then can lead to us hitting saturation points earlier or later. Now this next bit of material is taken in part from Chapter 1. It also is covered in Chapter 6 a little bit, but I'm going to pull some of this from Chapter 1, where we talk about homeostasis. So the whole point of homeostasis is to maintain internal stability. It was first kind of described by Walter Cannon in the 1800s. He was an American physiologist, and he's the one that coined the term homeostasis. And there's something that we should note here. Homeostasis, again, maintaining internal stability, you can think about it like we want to regulate, it's how we regulate the body's internal environment. And if we break this apart, homeo is going to mean similar, right? Meaning like or similar. And stasis means condition. So we're keeping a similar condition. However, it's not necessarily referred to as the same. So just like the ICF and the ECF aren't the same, you can have a homeostasis in which we keep different concentrations of solutes in the ICF versus the ECF. And that can be necessary to keep other things in line. So sometimes a better term we might use for homeostasis is homeodynamics. Like there's this dynamic change. It's not always steady. There is some change that occurs, some dynamicism. Why do we want to have constancy? What's the point of homeostasis? Well, we want constancy because our bodies, they function at ideal set parameters. So we keep, for instance, we keep a fairly constant internal temperature, 98.6 degrees Fahrenheit, right? What's the purpose of that temperature? Well, most of our cellular functions, respirations, and enzymatic reactions have an optimal temperature that they perform at, and that's around that optimal temperature. Ideally, that temperature is at a temperature that prohibits things like fungus and bacterial overgrowth from potentially occurring in some cases. So our body wants to keep things normal and constant so they can continue to function at its optimal level. So what happens when homeostasis is lost? Well, if the point of homeostasis is so that we can have normal functioning and optimal functioning, when we lose homeostasis, we have non-optimal functioning. And what we call that is disease, right? We have a pathology, a pathological condition in which now our internal environment is not at its optimal parameters. And what can cause that? Well, it could either be an internal pathology, or it could be an external in origin. So either one of those states, like let's say we're talking about body temperature, if we go outside and it's very, very cold outside, that can cause us to start losing heat. As we lose heat, we're going to try to attempt to compensate for that heat loss, and we have a number of homeostatic mechanisms that get triggered to try to raise and elevate our temperature. If that succeeds, if we're able to raise our temperature back up, then guess what? We have wellness, right? If it fails, then we develop illness or disease. So this physiological attempt to correct and maintain homeostasis is what we'll be spending a large part of this course talking about. Okay, again, we need to remember though, this is a dynamic steady state. We're constantly moving in and out of our set points, and when that happens, that's not the same as saying we have an equilibrium, right? There's not an evenness on either side of this equation, but we maintain this dynamic steady state. Okay, so we're constantly moving back and forth, especially between compartments of ICS, ECF. It's homeostasis, not homostasis, meaning the same. And it implies, if we think about it as equilibrium, that implies that the composition of the body compartments is identical, which they are not. Our ICF and ECF ionic concentrations will always be different, right? We have that disequilibrium, and that disequilibrium is needed so that we can maintain homeostasis. We use these ion concentrations to develop membrane potentials and utilize membrane potentials for long distance communication, like nervous action potentials, which we'll talk about next week. So taking all that, bringing it together, let's talk about the control systems that maintain our homeostasis. Okay, we regulate our variables within their body along a normal range by control mechanisms. So whether we're talking about pH or temperature, the body has what we would term a set point or an optimal value that it tries to stay around. And the control system that maintain that are under either local control or long distance reflex control. But regardless of which one of these mechanisms are under, or if they're under both, all systems contain the same three mechanisms. We have an input signal, we have an integrating center, which is kind of like our controller, and then we have an output signal that gives us a response. So let's look at this and think about it in another way. Let's say we have an aquarium, and we have a fish in that aquarium, and that fish needs to live at a very specific temperature. If it gets too cold, it'll die. If it gets too hot, it'll die. In order to maintain that temperature, we have a thermometer that goes into that then goes to a thermostat, right? And then that thermostat comes in and has a heater that heats the water. Okay, we have first a stimulus. There is a disturbance or change that sets pathways in motion. In this case, the thermometer is sensing that stimulus. Let's say I throw an ice cube into the water, which makes it colder. Okay, that change in temperature is the stimulus, okay, and the sensor is the thermometer. So stimulus is temperature, sensor, in this case thermometer, right? It monitors it. It picks up and says, all right, I've detected a change in temperature. And so it sends an input signal, which we would refer to as afferent, right? It is sending a signal to the integrating center, which in this case is the thermostat. So we send this signal, goes to the thermostat, to the integrating center, and at the integrating center, now that control box looks at it and says, okay, look, are we at the optimal temperature? No, we've moved away. We're further from set point. If we've moved away from set point, we need to make an action. If we're at set point, nothing happens. It just says, hey, we're good, don't do anything. But as soon as we move away from that set point, and it detects that, then it sends a signal via what we'd call an output or an efferent. You can think of efferent for effect. So an efferent signal gets sent to the heating coil, right? And that heating coil, that's the target, and it gets turned on and it starts heating up the water, right? Which then sends a signal, and as soon as the water gets back to the temperature, it's going to turn off the heater. Okay, this is what we would call a reflex control or a response loop. Okay, so there's this feedback loop in current. The response then feeds back again as a new stimulus, goes through the whole thing, and this is how we maintain most of the body's set points. Okay, so we have this stimulus, sensor, an input signal, integrating center, output signal, and then the target or effector that elicits the response. Let's talk about these parts. All sensors, okay, regardless of what type of sensor it is, they have what is known as a threshold. This is the minimum stimulus that's required to activate a afferent signal. Okay, so we could, some sensors could be just a cell membrane or an intracellular receptor protein, which something binds to it, and when it's bound, as long as it's bound, that might be the minimum threshold in which it elicits a response. Or we could have what we call neural reflexes, in which we have sensory receptors that are either central or peripheral, that are being stimulated by electrical signals. Okay, they could be stimulated by chemical signals, mechanical signals, whatever, right? And they have a minimal threshold of which they trigger their responses. When we talk about neurons, we'll talk about what that threshold is. That would elicit a neural reflex, right? We're using the pathways are going to be sent along neurons, and they're going to be primarily electrically, we're going to have electrical signals that get converted into a neurotransmitter. Okay. Then we can also have endocrine reflexes. Now we're using hormones. In the case of an endocrine reflex pathway, okay, we don't have, the endocrine cell is both the sensory receptor and the integrating center. So what we don't have is, and I'm going to change this on the PowerPoints. I'm seeing it right here. We don't have an afferent sorry, I'm writing with my finger. We don't have an afferent signal. Why? Because both the cell is both the sensor and the integrating center. So it's just going to be triggered, and then it's going to send, if it needed, it'll send an efferent signal in the form of a hormone release, right? So that input signal, it's going to depend on what type of reflex we're talking about. If it's a neural reflex, we'll have an electrical or chemical input signal. If it's an endocrine reflex, we don't necessarily have an input signal that comes into the integrating center. The stimulus directly acts on that cell, and it's going to be the integrating center itself. Integrating centers and neural reflexes are going to be cells usually in the central nervous system that receive those afferent path or afferent signals, and then they make decisions. So the brain has lots of integrating centers. In an endocrine reflex, again, that integrating center is going to be the endocrine cell itself that receives the stimulus. And then the output signals in these two types of responses, the integrating center evaluates the signal, and if the stimulus necessitates a response, then in the nervous, we're going to send either electrical or chemical transmitted by an efferent neuron. And in an endocrine cell, you're going to have hormones that get transmitted. And again, this comes to that question, if all electrical nervous signals are the same, how does the body distinguish where a system needs to go? And in the case of a neuron, it's going to be because it's a direct line from that region of the brain, from that integrating center, to the actual target system itself. So we have this line-to-line coding. In the form of the endocrine, how our response is specified? Well, by the hormone itself. So hormones have specificity to specific receptors. So only cells with that specific receptor will elicit a response from that signal. The targets of nervous reflexes tend to be muscles. They could be endocrine or exocrine glands or tissues. From an endocrine reflex, these cells, the target cells of endocrine reflexes, tend to be cells that have the actual proper receptors for that hormone. And then the responses that we get can be cellular responses, or we could also have systemic responses. So I love this table and this figure from your book. It's in the study guide. I have the table put on there. But this kind of shows how we can have a simple endocrine reflex where we're only using an endocrine pathway. And it shows here we have this internal or external change. Okay, there's no input signal, right? There's just the stimulus per se. We don't have this receptor. We don't have this input. The cell itself is the integrating center, and we have an output signal or hormone, target, and response. We have a neural reflex where we have the internal change. There's a receptor. It sends a signal that goes to the integrating center that sends then an efferent signal to the target tissue, and we have a response. And then we can have what's called complex neuroendocrine reflexes, where we're using both neurons and endocrine cells. So it might start off with a nervous response, but then it goes to a endocrine cell, and so we have this second response. If we contrast what's the point of having these two types of reflexes? Well, they each have their pros and their cons. So a neural reflex, it's really specific. Each neuron terminates directly on the single target cell, and so we can have a very specific immediate response that isn't necessarily firing off in other areas. Whereas in an endocrine response, most of the cells of the body are going to be exposed to that hormone. So the response has to be dictated by which cells have those receptors. And so it's not saying that, hey, one is better than the other, but this is very specific. This can also be very specific, but you can see how a neural reflex can be much quicker than, say, an endocrine reflex, in which you have to circulate a hormone for a long period of time and make sure all those cells come in contact with it. The nature of the signal, these ones are going to be mostly electrical, and then they get converted into a chemical neurotransmitter, where these are all going to be chemical signals. Speed, neural reflexes are very fast. Endocrine, going to be a lot slower because you have to circulate and give time for that hormone to be released, circulated, and then bind. The duration, neural reflexes usually very short. Okay, if you need a longer duration, then it usually gets mediated by like neuromodulators. Endocrine reflexes, these last longer than neural responses. And then finally, how do we code for the stimulus? So every signal in a neural reflex is identical in strength. Action potentials all have the same strength. So the way we code for the intensity gets correlated by the frequency of signaling. So the more signals we have, the more intense the response will be. In the case of an endocrine reflex, intensity is correlated with the amount of hormones secreted. So the more hormones secreted, the more response we will have. Okay, this goes through again and just kind of looks at these. Again, talking about what the sensors could be, integrating center, and it gives another set of how we can see how these things work. How we have a simple reflex, a neurohormonal reflex, complex neuroendocrine reflexes, and a simple neuroendocrine reflex. So make sure you kind of understand what's going through. This is color-coded, great figure that makes sure you understand this response. Lastly, we need to talk about the feedback loops. Okay, so most of these feedback loops are what we call homeostatic, in that we have a relatively stable set point that we're trying to determine and get back to. And what dictates that that stability, and what is it? How does stability depend on this set point? Well, if we get out of that set point, then we're not going to be able to engage in those cellular functions at an optimal level. And we have three types of feedback loops that helped us maintain these set points and maintain stability. One is what we call a negative feedback loop. Okay, in a negative feedback loop, the response that we get from our either neural or endocrine response, it is going to remove the signal. So it's like counteracting it. In the case of our example that I gave with the water temperature, the response was a temperature drop, right? We got colder, the stimulus was the change in temperature and made it colder. The integrating center elicited a response that turned on a heater. That heater is in direct opposite of the stimulus, right? It's trying to elevate the temperature. We use this to stabilize the regulated variable and restore a normal state, okay? But negative feedback, it can't prevent the initial disturbance. Negative feedback loop can't prevent, the water from getting cold. Most of our feedback loops occur through negative feedback. So examples would be, pH balance, temperature balance. All of these things occur through negative feedback. That's by far the most common. Now we also have feedback loops that we call positive feedback. In a positive feedback loop, now what happens is we are reinforcing the stimulus rather than decreasing or removing it. So if a negative feedback loop is, like, let's say your foot goes on the gas and the car starts to go further, it goes faster. So the negative feedback then would have you put your foot on the brake to slow down. The positive feedback, it's like you start to go faster and then you push on the gas even more to go even faster. At the end, what do you want to do? Well, we want to stop. And if you go faster, it may seem counterintuitive, like that doesn't bring us to a stop. Well, if you go fast enough and then ultimately you hit something, then you do stop. Okay, so there is ways that can kind of occur. So we send responses that regulate the variable even further than normal. We escalate what's happening, but the only way we ultimately can stop is that there needs to be some type of intervention or outside event that stops that loop from occurring. The classic example of positive feedback loop would be childbirth. Okay, so during childbirth, the woman starts undergoing contractions. With each contraction, the uterus squeezes on the fetus and pushes it down against the cervix. That pressure on the cervix, there are sensors in the cervix that feel that pressure that then elicit a response that releases oxytocin as a hormone. That oxytocin binds to receptors in the uterus that causes greater contractions. So we're contracting, and as with each contraction, the fetus pushes more on the cervix, which causes another greater contraction. So contractions get stronger and stronger and stronger and stronger. Now, contractions aren't normal, right? That's not normal what you want to have. The body wants to stop contractions. But really what's most important is getting the fetus out. The fetus has gotten too large. It's time to deliver that fetus. If it stays in, the mother is going to die. So stronger, positive feedback gets larger and larger, stronger and stronger contractions until what happens? Well, the baby passes through the birth canal and is delivered. At that point, the stimulus is removed, right? There's no more pressure being pushed on the cervix. So we stop producing oxytocin and the contractions stop. Okay, that's one example of positive feedback. There's several others that we could look at. Last type of feedback loop is what we call a feed-forward control. And this, the response loop is actually started in anticipation of change. So now the body is anticipating some type of change, and we start to prepare for it. A great example of feed-forward control would be when we talk about our gastrointestinal physiology. So when we ingest food and we break down that food, the body prepares for that food, namely the uptake of glucose, by releasing insulin. There's a number of different ways that insulin gets released that trigger the release of that insulin. The function of the hormone insulin, though, is to have cells uptake glucose into them. Well, as soon as you actually start to ingest food, when you start chewing and swallowing and your stomach extends, so the stomach stretches, that stretch, there's stretch receptors, and we have compounds like GIP and other hormones that get released that tell the body, hey, start producing insulin, even though there's no glucose that has increased in the blood level concentration. That's the primary way that that insulin gets released, is an elevation in blood glucose concentrations. But this is a feed-forward control in which we're anticipating that as that food gets digested, glucose is going to increase. So we've already started to change. We've already started this feed-forward control mechanism to deal with the oncoming increase in blood glucose levels. As set points are maintained, we can ask ourselves this point of, like, do set points ever change over time? Ideally, these set points are the conditions in which our cellular functioning and physiology is optimal. But we can have external environments that change to where maybe the set point needs to become a little bit more variable. It can change over time. Our set points are actually influenced by normal biological rhythms. So even some of the hormones that we commonly use to maintain our physiological functioning, like cortisol or something, they have fluctuations during the day. We have normal biological rhythms. Different people can have slightly different set points based off their inheritance and their genetics, right? Or if you just become accustomed to certain conditions, you're going to change your set point. If you live at a higher altitude than somebody else, where the oxygen or atmospheric oxygen is going to be slightly less, or at least in the partial pressure when you inhale, you're going to have a different set point of what your blood oxygen level may be. Okay, these biological rhythms, they get regulated, and they're fairly predictable and get repeated patterns. So we see things like, if we look at body temperature, right, at night, your body temperature actually gets higher, and then it declines. It starts declining during the nighttime. As you sleep, it lowers and lowers and lowers. And then as you wake up, that's probably the lowest body temperature you'll have. And as during the day, it'll increase at its highest. And then again, you'll go to sleep. It kind of goes through this, right? I talked about cortisol levels. Okay, we have really low cortisol levels when we sleep. It peaks in the morning, early after shortly after waking up, and then it'll have a second peak right before we go to sleep to help put us in these. So these changing set points of what these concentration levels are, they're important for normal functioning. Those are what we kind of call our circadian rhythms. They're adaptive and they have functions for that. We can also change our set points through processes like acclimatization, in which we have a natural adaptation of a physiological process to our environmental conditions. So there's a difference between acclimatization and acclimation. They sound very similar, right? But acclimatization is natural. So let's say I take you go from here in Hawaii, where we're at sea level, and then you travel to Denver, Colorado, we're at a higher elevation, and you're used to exercising here, it's going to be difficult at first. You're not going to feel like you're able to breathe all that well as you exercise in Colorado, but given enough time, the body is going to have acclimatization. You're going to produce more red blood cells in order to carry more oxygen, and you'll get used to it, and you'll have a natural adaptation to that. That would be acclimatization. Acclimation is where we adapt, but it happens artificially in a laboratory. So we force it to occur. It's not really a natural thing. It's not what we would find, right? So you acclimatize. It usually takes a little bit longer. Acclimation can be a little bit quicker because you're forcing a more extreme. But the body can do both. We're able to acclimatize and acclimate. So that's it for Chapter 6. Make sure you go over the study guide. Look over the study guide. That you look over the vocabulary. You look over the learning objectives. Those are the things that you're going to get tested on for the most part. And then answer the questions and submit them online for credit.